This is the lecture for Chapter 6, Photosynthesis. Photosynthesis is a biochemical reaction that begins with two inorganic reactants, carbon dioxide and water, and then light energy is used to create the high-energy organic molecule, glucose, and also a waste product of oxygen gas. On Earth, there are many different types of organisms that can perform photosynthesis. There are bacteria, which are a type of prokaryote that are photosynthetic. There are also protozoa, the euglena, are photosynthetic, as are different types of plankton that you find in the ocean, and they're unicellular organisms. In addition, there is the algae. Multicellular algae are called seaweed. And then, of course, what we usually think of as the photosynthetic organisms, the plants. Photosynthesis is an incredibly important biochemical process for all living things on Earth. And especially it's important in the idea of the flow of energy. And the flow of energy is how energy moves through an ecosystem. Now remember the laws of thermodynamics. Energy cannot be created or destroyed. So all living things need to deal with energy that is already present and change its form. So in terms of photosynthesis, energy, the original form of energy, is solar energy that comes from the sun. And photosynthetic organisms are able to take that energy and in the process of photosynthesis, convert that energy, solar energy, into chemical energy, the glucose. And those are the photosynthetic organisms, which we also refer to as autotrophs. Auto is self, troph is feeder. So we refer to them as self-feeders because they are able to take the solar energy and create their own organic molecules. And that is very important for the animals, because animals, to get their energy, they have to ingest the glucose that was created by the plants, and then they are able to use that glucose to perform cellular respiration, which will produce energy or ATP for the animal cells. And because the animals need to ingest autotrophs in order to acquire their energy, they are called heterotrophs. Hetero means other, troph means feeder. So this is the flow of energy in any ecosystem, terrestrial or aquatic. The energy originally comes from the sun. The autotrophs are able to convert that light energy into glucose. And then the heterotrophs eat the autotrophs, taking that glucose and converting that energy into ATP by cellular respiration. Photosynthesis is also important for production of oxygen. So if you notice with photosynthesis, the main purpose is to convert solar energy into glucose, but oxygen is also released as a waste product. And so this oxygen is released into the air, and oxygen is vital for the survival of all animals. So if oxygen was not produced by the process of photosynthesis, animals could not survive. And just to reiterate that photosynthesis directly relates to the production of all the food we eat. So obviously with the plants perform photosynthesis, so any vegetable is a direct product of photosynthesis, but even the animal products of milk and meat, the animals require glucose from the plants in order to survive. So all of our food is produced basically because photosynthesis can occur. And again, this food is going to be used by the heterotrophs in the process of cellular respiration to produce ATP for the animal cells. Because plants are the organism we usually think of in relation to photosynthesis, I'm going to go through the basic structure of a plant in terms of how they acquire the two reactants that are needed for photosynthesis. And remember, the two reactants are water, H2O, and carbon dioxide, which is a gas. So the overall structure of a plant, such as a tree, is that you have roots in the ground and then of course leaves above ground. And the purpose of the roots is to draw the water into the plant. So the importance of roots is to acquire the water, and that's how plants acquire their water that they will need for photosynthesis. And then that water has to make its way up the trunk, up the branches, to the leaves where photosynthesis actually occurs. And this is done through the characteristics of water. Remember cohesion, that's water hydrogen bonding to itself, and then adhesion, where the water can hydrogen bond to other charged particles. And that's capillary action, serving to draw the water up from the ground all the way up to the uppermost leaves in the tree. Now for leaf structure, this is a diagram of leaf structure, and the leaves, this is where the plant is going to acquire the carbon dioxide, and this is done through the stoma. Stoma are pores on the bottom of the leaf, the underside of the leaf, and it allows for gas exchange. So that's how the carbon dioxide is going to enter, and it's also where the oxygen is going to leave once the oxygen is produced. So carbon dioxide goes into the stoma, carbon dioxide goes into the stoma, oxygen comes out. Now some other parts of the leaf, you have the cuticle, which is on the outermost part of the leaf, and often that is composed of waxes. And remember, waxes are a type of lipid that prevent water loss. And so this water is one of those reactants for photosynthesis, so the plant doesn't want to lose water. Vascular bundle. The vascular bundle kind of acts as a circulatory system, kind of like your blood vessels. The vascular bundle, this is how water reaches those photosynthetic cells. So again, that is using the cohesion and adhesion of water to move the water from the roots up to the leaf. Now, photosynthesis is actually going to take place in the mesophyll. The mesophyll, this is the tissue in between the two edges of the leaf, and the mesophyll has the photosynthetic cells. So these are the cells that will actually be performing photosynthesis. So water is provided by the vascular bundle to those cells, and CO2 is provided through the stomata. And all of these cells, in eukaryotic cells, the organelle that performs photosynthesis are the chloroplasts. Chloroplasts are the organelles that are found in eukaryotic autotrophs. And I talked about them before with the idea of endosymbiosis. So endosymbiosis is the hypothesis of how eukaryotes evolved from prokaryotes. And the idea is that early on in evolution, chloroplasts were free-living photosynthetic bacteria. And a larger cell phagocytosed them, and they developed a symbiotic relationship. And some of the evidence for this is the fact that the chloroplast has two membranes. It has its own ribosomes that measure 70S, and they also carry a small circular piece of DNA. And they replicate the way bacteria replicate. But for the structure of the chloroplast, you should know that it has two membranes. So it has an outer membrane and an inner membrane. Also, interior to the inner membrane, there are thylakoids, which are these round discs, membrane discs. And inside each thylakoid is a lumen. So that is a sort of a liquid center. And a stack of thylakoids are called grana. Granum is singular, grana is plural. And the other thing you need to know is that the stroma is the liquid surrounding the thylakoids. And in terms of photosynthesis, light is harvested on the thylakoids, on that membrane, and then glucose is produced in the stroma. The basic equation for photosynthesis is deceptively simple. So in this example of the basic equation, you have two inorganic molecules, low energy inorganic molecules, the carbon dioxide and the water, providing the atoms, the carbon, the oxygen, and the hydrogen, to produce the product, high energy glucose, so that has carbon, hydrogen, and oxygen, and this is a high energy molecule and a much larger molecule. So this is an anabolic reaction. This is an anabolic reaction, and it is endergonic. So in order to convert the carbon dioxide and water into this larger molecule with more potential energy, it's going to require a net input of energy, and quite a bit of energy. And where does that energy come from? It comes from the light. So that's basically what is happening. That light energy is going to be converted into potential energy, potential chemical energy in the glucose molecule. And in the glucose, it's just good to point out that most of the energy in the glucose molecule is found in the covalent bonds between the carbons and hydrogens. So that's where you find that potential energy. Now, to do this, the light energy cannot be directly incorporated into the glucose molecule. So that light energy first has to be converted into ATP. So this is ATP, readily usable energy for the cell. And remember the structure of ATP. It has an adenine, a ribose, and 3 phosphate groups. So some of that light energy is going to be converted into ATP. And also, you have the production of NADPH. NADPH. So NADPH, and this stands for nicotinamide adenine dinucleotide phosphate. And this is a molecule that we call an electron carrier. It's called an electron carrier, but it's also carrying energy. So light energy is going to be converted into ATP and NADPH, and then those two forms of energy are going to be used to produce the glucose. And of course, oxygen gas is the waste product of photosynthesis. So it's just a waste product of photosynthesis, and that oxygen actually comes from the water. So the oxygen that makes up H2O, that oxygen is not involved in this process, and it's released as a waste product. Photosynthesis is also a redox reaction. And remember, redox refers to reduction, and oxidation. And these two types of reactions always occur together. And remember, reduction is the opposite of what you think, but it's actually the gain. So it's the gain of electrons, and electrons don't travel by themselves. They often travel with a proton. And when you have one electron and one proton, that makes a hydrogen atom. So reduction is the gain of an electron, gain of a hydrogen atom, and often it is associated with the gain of energy. Oxidation is the opposite of that. Oxidation is the loss of an electron, and so it's the loss of a hydrogen atom, and it is also usually associated with the loss of energy. And in a photosynthetic reaction, you are going to have a reduction reaction and an oxidation reaction. So remember, when you are looking at the molecules, it's probably easiest to follow the movement of the hydrogen atoms. So in this example, carbon dioxide, CO2, it is going to be converted into glucose, C6H12O6. So it is gaining hydrogen atoms. So carbon dioxide is going to be reduced during the process of photosynthesis. And those hydrogen atoms have to come from someplace, and where do they come from? They come from the water molecules. So during this process, those hydrogens are going to be removed, and so that the glucose can be formed. And then if the hydrogens are removed, then that molecule is just left with oxygen, and so it is oxidized. So that's where you see the reduction and oxidation reactions. In photosynthesis, carbon dioxide is going to be reduced into glucose, and water is going to be oxidized into oxygen gas. And that also follows the energy. Carbon dioxide is going to gain energy as it is reduced into glucose. So glucose is going to have more energy than the carbon dioxide. And then water is actually not, it's going to lose a little bit of energy when it loses the hydrogen atoms. The process of photosynthesis involves two basic reactions. And the first type of reactions that are involved are called the light reactions. And they're called the light reactions because this is where the light energy is going to be harvested and converted into chemical energy, the NADPH and also the ATP. And in order to do this, water is required. So the reactant water is required for the light reactions. And as a waste product of harvesting that energy and converting it into chemical form, oxygen is the waste product that will be produced. Once that energy is harvested and converted into chemical form, then that energy will be used in the second process, which is called the Calvin cycle. And the purpose of the Calvin cycle is to take the reactant carbon dioxide and convert it into the product of sugar. So remember, this is an anabolic reaction. You are taking a smaller molecule, making something larger, and it requires a net input of energy. So where does that energy come from? It comes from the chemical energy that was generated during the light reactions. So for photosynthesis, you have the light reactions, which harvests light energy and converts it into chemical energy. And then you use that chemical energy in the Calvin cycle to convert carbon dioxide into sugar. In the rest of the lecture, I'm going to go over the light reactions in the Calvin cycle in more detail. But before I even start talking about the light reactions, it's important to review light and its different characteristics. So light has the characteristic of a particle and also a wavelength. And so basically what light is a little particle that is moving with a wavelength. And as wavelength increases, as the wavelength gets longer, the amount of energy decreases. So conversely, a photon or a particle of light that has a very short wavelength that goes up and down very quickly is going to have more energy than one that has a very long wavelength. Now, all the photons we know with all the different wavelengths that we can observe, we have put them together into the electromagnetic spectrum. And this is the electromagnetic spectrum, and it organizes the waves or the photons by wavelength. So over here are the longest wavelengths, and over here are the shortest. And also, if you're thinking in terms of energy, this has the least amount of energy, and this has the most energy on this side. And so these are all the different photons arranged by wavelength. And the one we are most familiar with probably is visible light, because this is what we can see. When you separate white light out, when you separate those photons out by wavelength, you get the rainbow. So this is the rainbow, and the way to remember the colors of the rainbow is by an acronym, ROY, G, BIV, and this is backwards. This is ROY, red, orange, yellow, and then G, green, BIV, blue, indigo, violet. So that's a way you can remember the colors of the rainbow, and that's organizing the photons by wavelength. And red has the longest wavelength, so it has the least amount of energy of visible light, and violet has the shortest wavelength, and so it has the most energy of visible light. And then you should just be aware of the electromagnetic spectrum, that infrared has a longer wavelength than red, Microwaves is longer than infrared, and radio waves is the longest wavelength that we know. And so radio waves have the least amount of energy. If you go the other direction, ultraviolet has a shorter wavelength than violet, so it has more energy. X-rays have a shorter wavelength than ultraviolet, even more energy. And then gamma rays have the shortest wavelength and they have the most energy. And another way to think about this is the more energy that a photon has, the more damaging it can be. So visible light is not very damaging at all to us, but UV light, if you're exposed to too much UV light, you can develop skin cancer. X-rays, you know that X-rays can penetrate into the body to take pictures of interior structures. And gamma rays, that's a type of radiation that I talked about back with isotopes. When isotopes break down and they release radiation, gamma rays, this is radiation poisoning, and this can damage your DNA. Pigments are very important in terms of photosynthesis, and it's important to go over the basic characteristics of pigments. Pigments are molecules that can absorb and reflect different wavelengths of light. And pigments, we're used to thinking of them in terms of colors. So for instance, somebody painting, they have paints with different pigments in them, they give different colors. And so just as an example of how pigments work with the different wavelengths of light, if you have something that is blue, a blue pigment, what that means is that it absorbs all the colors of visible light except for blue. Blue is reflected. And since blue is reflected, that's what comes in contact with our eye, and so we perceive this pigment as blue. If something is red, same basic idea, it's going to absorb all the colors of the visible light except for red. So except for red, it's going to reflect red, and that is what we're going to perceive by our eye. White, a white object is one that reflects all the colors. So I don't know what happened to this image, but all those colors, red, orange, yellow, green, blue, violet, all of them would be reflected back to our eyes. And so we perceive it as white because it's all the wavelengths mixed together. Something that is black, if something is black, that means it absorbs all the colors of the rainbow, so it absorbs all of them, so nothing is reflected back to our eyes. So black is basically the absence of color. So those are how pigments work. So they deal with absorbing and reflecting different wavelengths of visible light. Pigments are very important for the process of photosynthesis. And that is because pigments are the actual molecules that are going to absorb the light energy. So they absorb photons of different wavelengths. This is a diagram that shows the different pigments that are involved in photosynthesis in plants. And probably the one you are most familiar with is chlorophyll. And chlorophyll is associated with a green color. The other types of pigments are grouped together as carotenoids. So these are the carotenoids, and they are colors from yellow to red. This diagram shows the absorption spectrum for the different pigments involved in photosynthesis. And there are three basic types that we talk about in photosynthesis: chlorophyll A, chlorophyll B, and the carotenoids. Now, chlorophyll A and B, they appear green. And the reason they appear green is because they reflect green light. So this is the ability to absorb wavelengths. Chlorophyll A and B tend to absorb purple and blue light, and they also absorb in the red range, orange and red. So because they are absorbing the purple and blue and the red, they reflect the green, we perceive them as green. So remember, with the pigments, it's what is reflected is the color we perceive it. And then the carotenoids, they tend to absorb in the violet, blue, and green range, and then they reflect the yellow, orange, and red range. And so that's why we perceive the carotenoids as yellow to red because they're absorbing the blue to green. Now, if you notice in this overall spectrum, most of the absorption is in the violet, blue, and green range. And this makes sense because violet, blue, and green, they have the shorter wavelengths, which means they have the most energy. So it makes sense that plants are going to focus on the shorter wavelengths of photons because that's where they're going to get the most energy. Now, in terms of leaf color, so this picture shows you different colors of leaves. Most leaves, when we look at them during the spring and the summer, they are green. So you have a lot of chlorophyll that is in the leaves, but there are also carotenoids there. You just can't see them because most of the pigment is chlorophyll. But during the fall, when the leaves start to die, the chlorophyll breaks down first, and then you can see the carotenoids that are left. So that's why the leaves in fall turn yellow, orange, red, because the chlorophyll is breaking down first, but you still have the carotenoids that are there. The light reactions are the first type of reactions that are performed in photosynthesis, and the purpose of the light reactions is to harvest light energy and convert it into chemical energy, more specifically into ATP and NADPH. Now, the light reactions involve certain structures called photosystems. And these photosystems are a cluster of pigment molecules embedded in a membrane. And that membrane is the thylakoid membrane. And remember, thylakoids are those flattened discs that you find inside the chloroplast, and a stack of those thylakoids are called a granum. So the thylakoid membrane is a membrane. It has a phospholipid bilayer, so it is a selectively permeable barrier, and embedded in it you have hundreds of different photosystems. And the photosystems are composed of all these different types of pigments: chlorophyll A, chlorophyll B, and carotenoids. And how a photosystem works is here is a photosystem with all the different pigment molecules. When a photon comes and interacts with the photosystem, it's going to transfer its energy to an electron in a pigment. And then that energy is going to get transferred to another pigment. So that energy from that electron is going to transfer to another electron on another pigment. And it's going to continue getting transferred from pigment to pigment, that energy, that electron, until it reaches the reaction center. And the reaction center is composed of a chlorophyll A molecule. So it reaches the chlorophyll A that is in the reaction center, and then that high energy electron is going to transfer to a primary electron acceptor. So that is how a photosystem works. When the photon transfers its energy to the electron of a pigment, that energy, that transfer of energy, transfer of an electron gets passed along to different pigments until it reaches the reaction center, chlorophyll A. And then the electron, high energy electron, is transmitted to the primary electron acceptor. And this occurs constantly when the chloroplast is exposed to light, because many different photons are coming in and interacting with many different pigments, and all of those pigments are absorbing the light energy and funneling that energy down to that reaction center chlorophyll, and all of that energy is transferred to a primary electron acceptor. So I just mentioned that when the photon interacts with the pigment, it's going to transfer its energy to an electron in the pigment. And just to explain that in a little bit more detail, I'm going to review the subatomic particles and the electrons and how they relate to energy. So remember, in the basic chemistry lecture, talked about the basic structure of an atom. In the atomic nucleus, you have the protons and the neutrons that are there in the nucleus, and then orbiting around the nucleus are the electrons. And when I talked about the electrons, I talked about how the distance the electron is, how far away it is from the nucleus, indicates how much energy it has. And the further away the electron is from the nucleus, the more energy it has. So the first shell has the lowest energy since it is closest to the nucleus, and the third shell in this case would have the highest amount of energy because it's furthest away. So in terms of transfer of energy, if an electron in a low energy shell, like the first shell, if it acquires energy, it will be bumped up to a higher energy level, higher shell, a shell further away from the nucleus. So that's what happens to electrons when they acquire energy. They are bumped up to a higher shell, to a higher energy level. So what actually happens to the electrons that are in the atoms that make up the pigments is that originally they're in a ground state. They're in a low energy state. So here is the nucleus, here's an electron, and it's fairly close to the nucleus. When a photon comes in, it's going to literally hit the electron and transfer energy to the electron. And when it transfers energy to the electron is going to be bumped up into a higher energy level. So that electron is going to be bumped up into a higher energy level, so it is moved further away from the nucleus. And when that happens to an electron, we say it's in the excited state. So it is bumped up, it absorbed energy from the photon, it's bumped up to a higher energy state further away from the nucleus, and it's in an excited state. And if an electron has enough energy transferred to it, it can actually be so excited that it leaves orbit from the nucleus. So it can actually leave the atom. There are two photosystems that are involved in the light reactions, and they are called photosystem 1 and photosystem 2. And they were numbered for the order that they were discovered in. But once they discovered the photosystems, they realized that photosystem 2 actually occurs first and photosystem 1 occurs second. So as I go through and explain these photosystems, I'm going to start with photosystem 2 because that is the first photosystem in the process. Now, photosystem 2 is sometimes referred to as the water splitting photosystem because that is part of the process. All right, for this photosystem, the main purpose is to harvest light energy. And light energy is in the form of a photon that is moving in a wave, and it's going to interact with pigments in the photosystem. So you know the photosystem is actually a big complex of chlorophyll A, chlorophyll B, and carotenoids. So as the photon interacts with one of those pigments, the photon is going to transfer its energy to an electron on the pigment, and then that energy, that electron, is going to get transferred to other pigments. And it's going to be transferred through the photosystem until it's funneled down to the reaction center chlorophyll, which is a chlorophyll A. And once it is transferred to that chlorophyll A, this electron has had so much energy transferred to it, and it's in such an excited state that it's going to leave the chlorophyll A and it's going to transfer to an electron acceptor. So an electron acceptor is an actual molecule. So that electron is going to leave that reaction center chlorophyll, be transferred to an electron acceptor. Once it is transferred to the electron acceptor, then that electron is going to be transferred to the electron transport chain, which is a series of proteins. So it's going to be transferred from one protein to another, and that's the electron transport chain. And during this process, that is where ADP is going to be converted to ATP. And that process is called chemiosmosis, which I'll talk about in a minute. So for the water splitting photosystem, the photon is going to transfer energy to an electron on the pigments. That electron gets filtered down to the reaction center chlorophyll, which is a reaction center chlorophyll A. That electron will have so much energy, it's going to leave that reaction center chlorophyll and be transferred to an electron acceptor, which is a molecule. Once it's gone from the electron acceptor, it's going to be transferred down a chain of proteins called the electron transport chain. Now, because these chlorophylls in the photosystem are losing electrons, they need to have those electrons replaced. And that is the role of water. So it's called a water splitting photosystem because when water is split, you're going to produce oxygen gas, and the hydrogen atoms are going to lose their electrons. Their electrons are going to be used to replace the electrons that are being lost to the electron acceptor and the electron transport chain, and then the hydrogen ions are left. Remember, a hydrogen atom is a proton plus an electron. So if a hydrogen atom loses its electron, we refer to it as a hydrogen ion. It's just one proton by itself. All right, so the purpose of splitting water is to replace those electrons that are being transported to the electron transport chain. And a very important part of photosystem 2 is how the electron transport chain produces ATP. And I mentioned that this process is called chemiosmosis. And this involves a membrane and different proteins embedded in that membrane and a process to produce ATP. This figure shows the process of chemiosmosis. So this is a diagram of a thylakoid. So this is the thylakoid, that membrane disc. This is the membrane, thylakoid membrane. This is photosystem 2 embedded in the membrane. And it's very important to know the different parts, that inside this membrane is the thylakoid space and outside of the membrane is the stroma. In the process involved in photosystem 2, so this is photosystem 2, the photon comes in, the light comes in, interacts with pigments in the photosystem, exciting the electrons in the photosystem. And once those electrons are completely excited, they get transferred to an electron acceptor. So an electron acceptor in the photosystem, and then that electron is transported to the electron transport chain. So this is the electron transport chain, involves different proteins embedded in the membrane. And one of the major membrane proteins in the membrane is plastoquinone. This is plastoquinone. So the electron gets transported from photosystem 2 along these proteins embedded in the membrane. And PQ, plastoquinone, actually also acts as a proton pump. So this is a form of active transport. And remember, active transport is where a protein carrier is required to transport molecules from an area of low concentration to an area of high concentration. So as this electron as the electrons are transported along the electron transport chain, this gives the PQ enough energy to transport hydrogen ions from the stroma, which has a low concentration, to the thylakoid space, which is going to have a high concentration of hydrogen ions. So movement of electrons across the electron transport chain is allowing active transport of hydrogen ions into the thylakoid space. So this is going to result in a buildup of a high concentration of hydrogen ions in this inner space. And that is important because the ATP synthase, this is a protein that produces ATP, it is going to allow the hydrogen ions to diffuse through the ATP synthase to go from an area of high concentration to an area of low concentration, which is in the stroma. And as the hydrogen ions diffuse through the ATP synthase, that is going to power the ATP synthase to take the ADP and attach a third phosphate to it, producing ATP. So this is the process of chemiosmosis. So chemiosmosis, the electron transport chain, movement of electrons across the transport chain, powers the proton pump to pump hydrogen ions into the thylakoid space. That high concentration of hydrogen ions in the thylakoid space are going to diffuse through the ATP synthase, so move from an area of high concentration to an area of low concentration, and that movement of hydrogen ions is going to power the ATP synthase to produce ATP. Photosystem 1 is the second photosystem involved in the light reactions, and this photosystem is sometimes referred to as the NADPH producing photosystem because that is the end product of this photosystem. So this is also a photosystem, so you have a complex of different pigments embedded in the thylakoid membrane, so complex of chlorophyll A, chlorophyll B, and carotenoids. When the photons come in and interact with those pigments, the photons are going to transfer their energy to electrons on the pigments. Those electrons get transferred from pigment to pigment until it gets to a reaction center chlorophyll. And then the electron is so excited it's going to leave the chlorophyll and go to an electron acceptor. So a molecule that is an electron acceptor. And then that high energy electron is going to be transferred to an NADP molecule. So that energy is going to be transferred to an NADP molecule. And remember, electrons don't travel by themselves. They often travel with a proton. And so one electron plus one proton creates a hydrogen atom. So what will happen is that the NADP is going to gain an electron, gain a hydrogen atom. So it's going to be reduced. NADP is reduced into NADPH. So it's gained an electron, gained a hydrogen, gained energy. So this is going to be a high energy molecule. I went through photosystem 2 and photosystem 1 individually, and now I want to put these two processes together to show you how the light reactions take light energy and convert it into chemical energy into the form of ATP and NADPH. So the first photosystem is photosystem 2, first photosystem in the process, water splitting photosystem. And what happens here is that the photon comes in, interacts with the pigments, transfers its energy to electrons in the different pigments. Those electrons ultimately become so excited they leave the reaction center chlorophyll and they are transferred to an electron acceptor. Then those high energy electrons are passed down the electron transport chain. As those electrons are passed from protein to protein to protein, that allows for chemiosmosis to occur, so that transport of hydrogen ions into the thylakoid space, active transport, and then allows the passive transport of those hydrogen ions through the ATP synthase, which converts ADP into ATP. So that's one of the major products of the light reactions. Photosystem 1 is the NADPH producing photosystem, and photosystem 1 is very similar. The photons come in, they transfer their energy to electrons on those pigments in that photosystem. The electrons get so excited they leave the reaction center chlorophyll and they go to the electron acceptor. And then those high energy electrons are transferred to the NADP molecule, forming NADPH, which is a high energy electron carrier. Now notice there's all movement of electrons, and overall through these two photosystems, there's a continual flow of electrons. So I mentioned in the water splitting photosystem that these electrons need to be replenished in these pigment molecules because they're leaving to be transferred to the electron acceptor and to flow through the electron transport chain. So new electrons to replace those that leave, come from water. So when water is split, oxygen gas is released, and that's the waste product of photosynthesis. And then electrons are moved from the hydrogen atoms, leaving protons, which are called hydrogen ions. So the electrons come from the water, transferred to the chlorophyll, and then as they're excited by photons, they move up to the electron acceptor, down the electron transport chain. After the electron transport chain, those low energy electrons, because they lost energy as they're transported down, are transferred to photosystem 1. And so now those electrons replace the electrons in photosystem 1 that are being excited by the photons, leaving the pigments, going to the electron acceptor, and eventually ending up in an NADPH molecule. So the flow of electrons through these two photosystems, the electrons start with the water, they move through photosystem 2, down through the electron transport chain, they go into photosystem 1, they're excited by the photons, go to the electron acceptor, and ultimately end up in an NADPH molecule. And so this is why water is required for photosynthesis. It's required to provide electrons for the light reactions for this whole process. And again, the waste product of splitting water to remove those electrons is going to be oxygen. After the light reactions, which occur in the thylakoid membranes, have harvested the light energy and converted it into ATP and NADPH, now it is time for the carbon dioxide to be fixed into sugar. And that process is called the Calvin cycle. And this occurs in the stroma of the chloroplast. This is a very simplified diagram of the Calvin cycle, and it essentially shows you what goes into the Calvin cycle and what comes out of the Calvin cycle. So the overall purpose of the Calvin cycle is to convert carbon dioxide into glucose. And in order to do this, this is an anabolic reaction. It's going to require a net input of energy, and quite a bit of energy goes into converting carbon dioxide into glucose. And it's going to require ATP and NADPH, so those are the chemical forms of energy that were formed during the light reactions. So this energy goes into this process, and then that energy is used to convert the carbon dioxide into glucose. And this is called a cycle because it's a cycle of enzymatic reactions, and a basic subunit gets recycled. So that's why we call this the Calvin cycle. This is a more detailed diagram of the Calvin cycle, and it shows the three different steps, which are called carbon fixation, reduction of carbon dioxide, and regeneration of ρBP. And remember, the overall process of the Calvin cycle is to take this carbon dioxide and create glucose. For this process, the first stage is carbon fixation. And carbon fixation basically means to get that carbon in the carbon dioxide into an organic molecule. And to do this, you need the enzyme Rubisco, also known as ρBP carboxylase. And the organic molecule that carbon dioxide is going to be attached to is called ρBP. So the Rubisco is going to take the ρBP, the carbon dioxide, and attach them together. And when it does this, it creates a 6-carbon molecule. And the 6-carbon molecule is very unstable, and it will quickly break down into 2 3-carbon molecules. So it basically breaks in half very quickly. So that's carbon fixation, getting that carbon dioxide into an organic molecule. Once that carbon dioxide is in the organic molecule, then it needs to be reduced. And remember, reduction as in oxidation reduction. Reduction is the gain of electrons, the gain of hydrogens, the gain of energy. So now you're going to have these 3-carbon molecules, you're adding energy to them. And that energy comes from the ATP and the NADPH that was generated in the light reactions. So those 3-carbon molecules are going to be reduced by ATP, reduced by NADPH, and eventually you're going to form this G3P molecule, glyceryl aldehyde 3-phosphate. And this is a high energy organic molecule. So high energy organic molecule, and this G3P can be used to produce glucose and other molecules. Now this is carbon fixation and reduction of the molecule, so you've ended up with the end product. But because this is a cycle, you need to regenerate the RuBP so this process can occur again. So now you're taking some of those G3P molecules and converting them into RuBP so the process can occur again. And to do this, this takes some energy also. So it's going to require some ATP that was also produced during the light reactions. And so this is the regeneration of RuBP. So you've replaced that RuBP, and now the process is ready to occur again. So if more carbon dioxide is available, more ATP and more NADPH is available, then this process will continue and more G3P will be produced, and the photosynthetic organism can produce glucose and other molecules. Once that G3P molecule has been produced by the Calvin cycle, that molecule is a high energy organic molecule. Now the autotroph, the photosynthetic organism, can make all sorts of different types of organic molecules. So remember, in metabolism, I showed you a biochemical map of all the intertwined biochemical pathways of an individual cell and how cells can regulate and control their pathways. That is the same for an organism. So this autotroph, which made its own organic molecules, it can now take that G3P and it can use it for amino acid synthesis if it needs to. Amino acids are the building blocks of proteins. So if it needs to make enzymes, it has the ability to do that. G3P can also be used for fatty acid synthesis. Fatty acids are a type of lipid, and in plants, this would result in oils probably for the plant. So vegetable oil, olive oil, corn oil, and fatty acids or lipids are used for long-term energy storage. Most of the G3P is probably converted into glucose. And this glucose can be complexed with fructose to form sucrose. And sucrose is often in plants a transport molecule. It's found in the leaves, the fruits, and the seeds. Or the glucose can be linked together to other glucose molecules to form starch. So that's energy storage in the roots and seeds. Or the glucose can be modified and converted into cellulose, another type of polysaccharide, which is used for structure. So again, this is why photosynthetic organisms are referred to as autotrophs, self-feeders, because the process of photosynthesis is going to allow them to produce G3P, and from that molecule they can produce any type of macromolecule that they need. C3 plants are plants that are adapted best to moderate environments, because if the environment gets too hot or too dry, they are prone to wilting. They lose water very easily. So to prevent water loss, they close their stomata, so to prevent the water from leaving the plant, but when they do this, this prevents the gas exchange. So it prevents the plant from acquiring carbon dioxide, so it reduces the ability of the Calvin cycle to function, and it also increases the concentration of oxygen in the leaf. And so these are plants that do not do very well in hot or dry environments, and they're plants such as wheat and rice and oats. C4 plants are better adapted to dry and hot environments because they have a different cell arrangement that allows them to continue the Calvin cycle even when the stomata are closed. And because of this different cell arrangement, they have bundle sheath cells that can store carbon dioxide in a molecule that has 4 carbons. That's why it's called C4, or they're called C4 plants. So these cells can store carbon dioxide so that when the stomata close, there's still a source of carbon dioxide for the Calvin cycle. The last type of plant are CAM plants, and CAM plants prevent dehydration by opening their stomata only at night. So at night, they open their stomata so they can have gas exchange, and during that time, they fix their carbon dioxide into the C4 molecules. So during the day, when they close their stomata to prevent water loss, they still have a source of carbon dioxide for the Calvin cycle. So CAM plants will only open their stomata at night for gas exchange and then close them during the day to prevent water loss. And examples of CAM plants are succulents, like aloe vera, and also the pineapple plant. This is the end of the lecture for photosynthesis. Photosynthesis essentially is how autotrophs can take carbon dioxide and water. Using the energy from light, they can convert them into glucose and the waste product of oxygen gas.